Well, good morning. My name is Simon Loveday. I'm a senior scientist at AgResearch in New Zealand. I'm sorry I can't be with you in person, but I'm currently asleep. It's 2 a.m. in the morning in New Zealand, but here's a recording for you. So, topic today is, could dairy factories process plant proteins, pivot to plant proteins? Just to give you a bit of background about myself, my PhD was sponsored by General Mills on fresh and frozen dough biochemistry, and then I moved into the dairy protein functionality as a postdoc. And about 5 or 6 years ago, I came to the realization that a whole lot of new proteins are coming along. We need to understand them. We need to understand the opportunities, whether they present threats or opportunities to the dairy industry. And so I started having a look at dairy hybrids, things like pea and hemp and soy and quinoa proteins. And today I'll talk a little bit about some of the differences between dairy and plant proteins and whether we might be able to process plant proteins in dairy factories. First question is, why would you want to pivot dairy factories to plant proteins? In the New Zealand context, our dairy production is very seasonal. About this time of year, most of our cows are dried off because they're outside in the pasture and the grass growth is slow during the winter months. And we have excess capacity in our dairy factories in the winter. And so various people have raised the idea that could dairy factory excess capacity be filled with plant protein processing in the seasonal system? In the wider context, a lot of people are talking about plant proteins. Excuse me. There's rapidly increasing demand. The industry is growing fast, and so there could be an opportunity for some profitable pivoting there. So, speaking of profitability, would it be profitable to process plant proteins in dairy factories? I don't know, but let's do a thought experiment. Let's have a look at what would we have to do to process plant proteins in a dairy factory. If we wanted to do that, how would we do it? Follow me down the rabbit hole and let's have a look at this this interesting question. I wasn't sure whether Americans use the term rabbit hole, but Merriam-Webster tells me that you do. So let's we're on the same page. Okay, first question around processing plant proteins is, which crop are we looking at? And broadly speaking, the main protein-rich crops are the oil seeds, the legumes, and the cereals. And they're all different. If we look at the composition of different crops, so we have some oil seeds, we have a legume, pea, we have quinoa, which is a what is it? Edible seed, technically speaking. And we look at, here's a plot of the dry matter content of protein, oil, carbohydrates, ash, and fiber. And they're hugely variable. Hemp has can have more than half of its dry weight as oil. Pea is mostly starch and protein. And so very different composition to start with. And as you'd expect, we'd process these differently. In particular, the oil seeds that are very high in lipids, we know that lipids interact with proteins in ways that make them difficult to separate. And so oil seeds require defatting before protein extraction. A brief tour through plant protein chemistry. In the plant protein world, the classification revolves around the albumins, globulins, prolomins, and gluteolins based on the solubility in different solvents. The albumins soluble in water, globulins in salt solution, prolomins in alcohol, and gluteolins in dilute alkali. And some examples down the bottom there. If we look at the cereals, they tend to be rich in the prolomins and gluteolins, very low solubility in water-based solvents. The legumes have predominantly albumins and globulins, and so they're a bit more amenable to water-based protein extraction. Speaking of solubility, here's some graphs of the pH on the x-axis versus the solubility on the y-axis. And a number of plant proteins have this distinctive minimum around acid pH, 4 to 6, similar to casein. and it tends to be a bit of a broader minimum. The position varies a bit, but it's a similar pattern. And so this gives rise to some ideas of, can we process these proteins in a similar way to how we handle casein in dairy factories? Well, stay tuned. The poster child for dairy protein, for plant protein processing is soy. The industry's been around a long time. The vast majority of soy beans are processed for oil, firstly, and for meal for cattle feed, secondly. A tiny fraction of soybeans are processed for edible soy protein ingredients for human foods. But we can learn a lot from the soy industry and the processes used there. Broadly speaking, there are two parts to the process: preparation, which is pre-processing and conditioning, and then separation and refining. And we'll have a look at those in more detail in the next few slides. In the pre-processing and conditioning side of things, soybeans from the fields have to be dried in order to be stable and tempered to the right moisture content. Then they're cleaned. You get a lot of dirt and stones and non-target seeds and bad seeds that are removed. Seeds are cracked, dehulled, so the outer husk is removed, conditioned, flaked, expanded, and then you have collets or pellets or flakes ready for further extraction. So a number of unit operations happen in this pre-processing stage: sieving, aspiration, cleaning, sorting, tempering, scouring, flaking, and so on. And the kind of equipment used to do this, here's a Buehler color sorter. Here's a dehuller for breaking off those outer hulls. And here's an expander for flaking or colleting the seeds. And do we see any of this hardware in a dairy factory? No, we don't. So that's our first clue as to whether we could do this in a dairy factory. Following that pre-processing stage, we have oil separation by solvent extraction. So those collets come in from the expander. Hexane is added. You can use other solvents, but hexane is the most efficient in terms of extraction efficiency to cost. The oil is dissolved in the hexane, and the non-oil material is then passed through a desolventizer toaster, dried and cooled for soy meal. And various processes happen to the oil as it's removed from the hexane. And again, the kind of hardware involved in this process: DTDC for the processing the meal, solvent extractor by the rotocell process or the loop type extractor, and then all the hardware associated with refining, bleaching, and deodorizing the oil. And again, we ask the question, do we see these in a dairy factory? No, we don't. So this oil separation phase is problematic as far as using a dairy factory. Then once we have defatted meal, we're looking at the extraction and concentration of proteins. And this is a different story because a lot of the processes here are familiar to the dairy industry. This is a plant protein concentrate process. So the defatted flour comes in here, acid is added, the soluble material is removed, and the protein is insoluble in this acid pH. So here's a plot of pH versus solubility for soy protein. We're in that deep minimum in acid pH where the solubility of protein is low. So it's in the solid phase. That's washed and processed and dried. That's a protein concentrate process. And about 70% of the protein is recovered from this concentrate process. If we move into the alkali region where solubility is high, the protein dissolves in the alkali. Here's a protein isolate process. Again, that defatted meal or flour comes in, alkali is added, and this time the protein dissolves in the liquid phase, the aqueous phase. The solid phase is where the fiber and the starch sit. A separation of that solid from liquid gives you an alkali solution of protein. That protein can be either dried directly or concentrated by acid precipitation or ultrafiltration or other processes, and spray dried into a nice food ingredient powder. Here's an example of a plant protein isolate process flow diagram from GEA. So you have a pH adjustment tank, the flour, the meal, and the alkali added, decanting stage separating the dissolved alkaline solution of protein from the solids. Second decanting stage, the dissolved alkali solution goes into a pH adjustment tank, precipitation happens, and then you have, again, a decanting separation and a drying stage. Fiber and starch are processed in other flow streams, and then you have effluent processing. Here's an acid casein process, and you'll see that some of the hardware is remarkably similar to this previous slide on a plant protein isolate process. So the casein is acidified, and then we have decanting and washing stages. One of my colleagues pointed out that you tend to have a screening stage before the decanter, and you might have only one decanting stage. But we have similar hardware in this acid casein process to the plant protein isolate process. Decanters, pH adjustment, washing, pasteurizing, and then drying and packaging. So we see pH adjustment tanks, we see decanters, we see spray dryers, we see effluent treatment. One question I still have is whether fiber and starch could be processed in a dairy factory, and that's an open question. So in summary, here are the various processes involved in making plant protein ingredients, and the question of whether they could be done in a dairy factory. Those first two stages of pre-processing and defatting, probably not. Extraction with acid, alkali, or salt, maybe. And then those protein extraction, concentration, precipitation, and drying stages, yes, they could definitely be done in a dairy factory. Here's a quick detour to look at protein ingredient functionality. So here's a range of different materials, protein-rich materials extracted from different crops. Here's the solubility. On the top, we have whey and casein. And what we notice is that dairy protein solubility is extremely high compared to other plant protein materials. Plant protein solubility is extremely variable, from very poorly soluble right up to similar to a whey protein isolate. And this points to the fact that the functionality of the ingredients is very dependent on the process, on the crop, and we can modulate functionality by our choice of processing. One of the problems that leads to low solubility is that the defatting process involves heating in the expanding flaking stage, and that damages the protein and leads to cross-linking aggregation, poor solubility. One of the ways around that is cold pressing. So here's a cold pressing, doesn't involve heating, obviously. Here's an example of a small cold pressing operation. This is from a hemp production facility. This one, Kanapu hemp seed oils, located in Otane, not far away from the place behind me, beautiful Hawke's Bay in New Zealand. And this process produces a seed cake that has about 12 to 15% oil in it. It's still a fairly high oil material. However, we have some tools at our disposal to deal with that oil content. The other problem that leads to low solubility is the acid precipitation step can lead to cross-linking and make it hard to redisperse the protein. However, we know all about whey protein isolates and the fabulous solubility we can get by ultrafiltration. So here's a potential solution. Here's a whey protein isolate process. I've bolted on some of the steps from the plant protein isolate process at the start. And here's how we could deal with that. Cold pressed cake that has high protein functionality, but some residual lipid content. So those first stages of alkali extraction, decanting, could then feed into a whey protein process where we have a separation stage, the fat's removed, then an ultrafiltration stage to concentrate the dissolved protein, a microfiltration stage to remove any residual fat and some microbes, and another ultrafiltration and diafiltration if we really want to clean up to a very pure protein isolate. So the point I'm making here is that this whey protein isolate process could be quite well suited to processing cold pressed oilseed cake and produce an excellent functionality in terms of avoiding the damage to the protein that results from heating and the insolubility from the acid precipitation stage. Here's another process for producing really soluble protein. This is patented by Burcon in Canada, and you might have heard about Merit Functional Foods canola protein ingredients. This is the same. This is Burcon's the parent company of Merit, and their process uses salt to extract the globulins from rapeseed or hemp. Canola. So a salt extraction process. This takes advantage of the salting phenomenon of certain proteins. And then we have a washing and concentrating UF and DF process. Again, very familiar kind of hardware from the dairy industry. The key difference here is that the protein is concentrated by rapidly dropping the ionic strength. You get what's called a micellization process, and you get protein micelles formed that retain very good functionality, very good solubility, and yeah, it's another way to achieve good functionality from plant proteins that are inherently different from dairy proteins. So to come back to the question, could a dairy factory pivot to plant proteins? Yes, with a little help from our friends in the arable sector. And here's a schematic of how it could work. So the seeds, beans, or nuts come in from the farm. That pre-processing and defatting requires quite specialist equipment, which we don't see in a dairy factory. But the defatted meal could then be subjected to protein extraction and fractionation using existing dairy processes, dairy hardware, and to produce a range of fractions for food ingredients. Coming back to the definition of a rabbit hole, Merriam-Webster points out that this is a complex or bizarre situation that can lead to other questions, problems, or pursuits. And as you'd expect, nothing is as simple as it looks in 15 minutes. So some further questions that we might want to pose in this question. Would the food safety regulations permit plant materials to be processed in a dairy factory? Could those secondary streams of starch and fiber be processed on existing dairy hardware? And would it be profitable to follow through that joint venture process where the arable industry does the first part of the process and then the dairy industry can do that protein concentration extraction? The answer, I don't know, but thank you for joining me to have a look down this rabbit hole of could dairy factories pivot to plant proteins. I'm sorry I can't answer your questions. I'm still asleep. It's about 2:30 a.m. in New Zealand, but I'm very happy to hear from you by email, LinkedIn. Yeah. Here are some quick references. You can pause the video if you want to look at those. Finally, thank you to the organisers for the invitation to speak, and I hope you have a fantastic symposium. Thanks very much. Goodbye.